everyone. Welcome to our Alzheimer's webinar. I'm Chris Lindgren with QMENTA. Just a reminder for folks, if you have a question today, please use the Q&A function at the bottom center of your screen. That way our moderator will be sure to see your questions. Please don't use the chat for questions. We do plan to take questions at the end of our panel discussion today, so please submit them. And I'm now going to turn it over to Glenn Thomas, QMENTA CCO and our moderator.
Great, thank you so much, Chris. Good morning, good afternoon, good evening, everyone, wherever you are. Welcome to this, our latest in our series of QMENTA webinars. And we have a cracker for you today. The Aduhelm FDA Approval, Fresh Insights and Opportunities. My word, in the last 3 months or so since the FDA approval for Biogen's Alzheimer's disease therapy, we've seen resignations, heated debate, a label clarification, an ongoing congressional investigation. And I don't know about you guys, but I can't open a news feed without some new development or commentary related to this. But with this being the first Alzheimer's disease therapy approval in like nearly 2 decades since Razadine, and with the FDA seemingly adopting a more agile mindset, this approval has, I think, above all things, given hope to Alzheimer's disease and other victims of neurodegenerative diseases around the world. Among the discussion and debate about Aduhelm itself, I think we really believe that what's most important is the lessons that we learn from this and how we can build on the positives from the FDA approval. So, what has made the approval so significant, and what are the opportunities that exist for the future of clinical study execution in Alzheimer's disease and beyond? Well, we have an outstanding panel of academic and industry experts on hand here to unpack and explore this fascinating topic. Each of our panelists is a scientific PhD, a medical doctor, a combination of the two, all of which really make me question what I was doing with my life earlier on. So let me go to each of the panelists and perhaps ask them to briefly introduce themselves. Firstly, we'll start with Dr. Martin Tolar, PhD. Hi, Martin.
Hey, Glenn. Thank you for inviting me. So I'm a neurologist by training. I did my PhD on Alzheimer's. Actually, I worked on the ApoE4 when the connection to the disease came up. I trained in Boston, went to Yale on faculty, and then was really frustrated with the lack of therapeutics. I went to Pfizer, ran a number of programs there, then ran the business group, and then 4 biotechnology companies. First was a company called QMENTA. We were the first to bring beta secretase to clinic. Did a $1 billion deal with Astellas. It turned out that this is not the right approach, as we now know, and founded Alzheon 8 years ago. And now we have the only oral approach to target the toxic amyloid in late stage development. Pleasure to be here.
Great to have you, Martin. Let me introduce Duygu Tosun-Turgut. Duygu, do you want to say hello?
Yeah, thank you. So my name is Duygu. By training, I'm an electrical engineer and mathematician, but I found my way to neurology and neuroimaging during my PhD and my postdoc. Currently, I'm an associate professor at University of California, San Francisco, and I'm the director for medical imaging informatics and artificial intelligence. And a big portion of our research efforts is around biomarker discovery, particularly for neurodegenerative diseases. And we particularly focus on how to optimize clinical trial designs with these biomarkers, either to detect the diseases or monitor or predict the disease progression. And thanks so much for inviting me. I'm so happy to be here this morning.
No, it's great to have you, and you can tell from your introduction, your background is right on point for this topic today.
Thank you.
Juan Domingo, Gispert, PhD, my friend. Do you want to say hi to everyone in the audience?
Thank you, Glenn. Thank you, everyone, for having me today. I'm a group leader at BPRC, which is an academic institution focused on prevention of Alzheimer's disease. My background is also technical. I'm an engineer by training. I've been working with neuroimaging data for over 20 years now. I have been involved in anti-amyloid clinical trials for over 10 years. For basically amyloid PET imaging. Thank you.
Great to meet you, and great to have you on the panel. And finally, my dear friend and colleague here at QMENTA, Paulo Rodrigues. Paulo, do you want to say hi?
Yeah, no, thanks, and a pleasure to join like such a great group. So I'm a software engineer by background, and then I did my PhD in neuroimaging in the Netherlands, in fact, and it was really focused on neuroimaging, data engineering, so like how to analyze medical images, MRI, and other data, especially with the focus to understand like how diseases are damaging the brain, are damaging the tissue, and really towards like developing new imaging markers to understand it, to quantify, actually that's the word, to quantify damage in the images. So I heard also like a word that resonated with me, like the frustration sometimes in science and in some of these fields, right? Some of the slow pace and like in the translation of some of this technology to the market. So back in 2013, co-founded QMENTA, really with that mission to empower breakthroughs in neuro disorders. So we're here joining this very interesting topic that I think is really resonates with the fact that we built a cloud-based data analytics platform, really to bring these two points, right? Like how to streamline medical image management to really speed up and improve processes in clinical trials and clinical studies, but also with the angle on how to bring or how to give more accurate results, faster results, and with confidence. I think that's an important word also today.
Yeah, I think there's some things in there to do with when we unpack some of the stuff with the Aduhelm approval in terms of confidence of the data, of the surrogate endpoints and so forth. I think we'll perhaps get into some of that stuff over the course of the next hour or so. So hey, let's get into it. I think it's going to be really helpful just to get us grounded. And Duygu, I read a really great summary that you put together a while ago on this topic. So I'll ask you if you wouldn't mind. So tell us about this approval. What made it so unusual? What's it all about?
Yeah, thanks, Glenn. So in early June 2021, we had this approval, and agency did this through an accelerated path, which was originally designed to increase the availability of new drugs for serious and life-threatening diseases, for which we have very limited therapeutical options. And this is the FDA's accelerated approval pathway. And many of you might remember this, but this was originally used back in 1990s in the wake of HIV crisis. And since then, and especially in recent years, we're actually seeing more and more new drugs getting approved through this expedited, accelerated pathway rather than the usual traditional FDA's drug approval pathway. So in an accelerated approval, a drug can be approved not based on the evidence that it has a clinical benefit, which is usually the standard way how we assess drugs, if it is improving the clinical symptoms or the cognitive symptoms, but rather it relies on evidence that is that the drug is changing the biology of the disease in a way that it might likely have a clinical benefit. So these are the things we call surrogate endpoints, and these are like laboratory tests, radiological images, or other biomarkers. And the surrogate measures, these surrogate endpoints, are expected to have a reasonable and likely prediction of a clinical benefit. And I guess this is where it gets a little bit unusual for this drug. So in the case of Aduhelm, FDA's accelerated approval was based on their phase 3 trial results, which consistently show that the drug actually removes amyloid plaques effectively from the participants. There was a beautiful number, it's like 55 to 60% reduction on amyloid accumulation on average. But this is where it gets a little bit complicated, because when we think about Alzheimer's disease, it has all these overlapping and sequential biological changes, biological processes. And the way we conceptualize Alzheimer's is there's the upstream accumulation of amyloid plaques, oligomers, that leads to stative downstream accumulation of tau, synaptic dysfunction, neurodegeneration, which leads to cognitive decline and clinical dysfunction. So the controversy is whether removing amyloid plaques really benefits the patients in terms of cognitive and clinical dysfunction. That's a big question, even in research and as well as in clinical trials, especially Aduhelm is approved for symptomatic stages of the disease. This is way beyond the initial amyloid accumulation, and this is where we are in neurodegenerative state of the disease.
Fascinating. So as far as if you like, the controversy around something like this, you would say it's as much associated with the surrogate endpoint and whether that actually translates to true patient benefit and impact. Is that a fair way of summarizing the issue from your perspective?
I think so, because especially now we are considering this as the very first disease-modifying treatment for Alzheimer's disease that's been approved, the very first one that's been approved past 2 decades, and all the other options, medication we have for Alzheimer's is really symptomatic treatment. So this has this, honor and the label of being the very first disease-modifying disease treatment. It's important to have that true evidence that removing this pathology, this amyloid plaque, truly going to improve patients' symptoms. That's a fair question to ask.
Yeah, and I think with a terrible disease like this, where unfortunately the clinical endpoints experienced is often fatal. You can see, where people have wrestled with this. Other perspectives, Paulo or Martin or anyone else. What else about this approval kind of made it so controversial? Let's say.
I mean, let me just maybe go back a little bit history. So it was a very good summary from Duygu, actually, of I think one of the scientific crux point. But really the main controversy has been around the work. So it's kind of led to everything else. Has been the fact that the Biogen ran 2 phase 3 studies of about 3,500 patients, and they stopped the trials for futility, right? So they stopped it basically with 40% of the patients never completing the studies, because their calculations from their initial look at the data has been that there's just such a small chance that this is going to work, this is going to show positive data effect, that actually stopped the studies. So and then they reanalyzed the data, in fact, a few months later in the post-hoc analysis fashion, and then they found that in one of the studies, one of 2, there was a positive result. So that really led to what Duygu said. This was not sufficient for standard approval, even though the program and the data set otherwise would be, right? I mean, because again, they had a very large phase 3 program and so forth, and that's where they invoked the plaque, as Duygu very well said, which has been shown not to correlate with the cognitive impairment or improvement, removal of the plaque as a surrogate, right? So they basically set a precedent for plaque being used as a surrogate, with really very little support for that from all the data. In fact, we know that you can remove all the plaque from the studies with the other antibodies. The most probably compelling is what's called the GAP study with the Eisai's lecanemab, where they stopped the treatment and there was no plaque, in these patients, and they progressed exactly the same way as the patients who, did not have a treatment at all. So that really led to kind of this the plaque using as a surrogate. And then finally, the advisory committee who reviewed this voted unanimously against the approval. So they overturned that. And then finally, the other 2 controversies were the breadth of the label. Initially, FDA gave them basic label for all Alzheimer's that the Biogen itself actually asked, to give it just for the population they have studied, which is the early Alzheimer's. And finally, all the discussions about the pricing and so forth. But again, I mean, I would agree with Duygu that the most problematic probably on all of this is the attempt to use the plaque removal as a surrogate for efficacy, which we know from so many other studies is just not the case.
Yeah, it's interesting as well. When you look at some of the commentary, I was looking at a quote here, I think from the CEO of Biogen, Michael Van Attos, saying clearly too much confusion, misinformation, and controversy surrounding our data and the approval process has kind of contributed towards a lot of this. And I think that was one of my observations, was that just so many moving parts in this, including just that the way that the data was presented and analyzed, stopped, and then retrospectively submitted, created a lot of noise into this. But it has of course been very positively received by so much of the if you like, the patient community here, right? People desperate for something to address. I mean, Juan, do you have a perspective on what this approval, not just this approval itself, but this shift in direction with the FDA, with neurodegenerative diseases and Alzheimer's, what this means for patients?
We have relatively close contact with patients and patient associations because we have a network of over 50,000 associates to BBRC, to our research institution. And we have promoted some initiatives in the Spanish Congress, for example, with patients and so on. I think you summarized it very well at the beginning with one word, which is hope. Of course, Alzheimer's leaves little hope to patients, unfortunately. So it's devastating, and patients are looking for something that changes the situation. And in this regard, all patient associations have welcomed with open arms the approval of aducanumab. In the United States, you have a very powerful association, which is Alzheimer's Association, with a very strong voice. They even had this campaign before the FDA approved the drug. It was called More Time, and they engaged some celebrities like Samuel L. Jackson to raise the awareness of the FDA having to take this decision. And of course, they were very happy with how it turned out, and now they are also continuing. And in Europe, also we have a very different, the structure of patient associations is very different. Probably the voice is very much fragmented, but all of them unanimously have greeted the FDA decision. Alzheimer's Europe, for example, has publicly said that they are very happy that this has happened, and they also expect that the same thing will happen in Europe. And mostly what patients are concerned about is the eligibility. So at what stage of the disease that this might be useful, whether positive amyloid biomarkers will be required, where specific genetic groups will be allowed to take the drug, even not in the same disease stage, and also side effects. So how side effects, what are they, how severe they may be, and how they might be handled from a clinical perspective. Other important aspect is that patients' associations have a say in regulatory agencies. So in the advisory panel for the FDA decision was a representative of patients, which in this case didn't vote differently as the other members of the committee. But also in Europe, patient associations might participate in the EMA discussions or whether the drug is finally approved or not.
That's fascinating, and it's for me it's particularly striking, and I think we've talked about some of this in the past in some other webinars, that the general public have seen that that our community can move quickly when it comes to, let's say, vaccine productions in response to COVID-19, right? And they've seen that the world can rally around, and that regulatory bodies and governments can be more agile in the face of these diseases when necessary. And so then there was this question, I think, that came up, well, so will patients and will people have the patience for traditional long approval processes for awful diseases like this, where they see family members living with a life sentence as a result. And that voice of the patient in the process, I think it feels like it's been heard here, right? So I think that's fascinating to see that.
Also, I think that they're very realistic in the terms of what the drug can and cannot do. In the sense, I think we all agree that at best has a modest clinical benefit. So patients are aware that this is not the cure for Alzheimer's disease, right? So they're pretty aware of that. And also I think we have to be careful, and when we publicly speak about what these drugs cannot do, we have to be very prudent, because that actually affects many people's lives in a very deep way.
Yeah, 100%. I think there's a great level of responsibility for all of us as we're talking about this stuff. You spoke on just now about the patients. Martin, let me turn a question to you and others to add. What's been the reaction of the industry and its investors onto this? This has been pretty radical, right?
This has been tectonic, change. You cannot actually kind of overstate, how dramatic the change has been on the industry, on the investment community, and on everybody, because we were literally on a desert for decades, right? The investments in Alzheimer's in drug development were estimated at 600 billion. There was actually a Biospace article, 600 billion down the drain with nothing to show for it, which really led to exit of virtually all the big pharmas. Again, I think we should be grateful to the few that actually remained. And if I can have the first slide, we'll talk about it a little bit more. But again, I mean, the impact on the industry has been incredible, and really, allowed the investments, the really reignited the interest in all of the science and so forth, and had a tremendous impact all over the space. Now, what really, the main impact probably has been, sort of practical impact, going to what Juan was saying earlier, has been that there's true Alzheimer's pipeline. There's one approach that we know, which is targeting the toxic amyloid. There are a number of data sets that had positive data. It's the antibodies, the aducanumab we talked about, but also there are positive data from the studies with lecanemab, the Eisai's antibody, and also the donanemab release antibody. So there, and ours, which is the oral approach to preventing the forming of these toxic aggregates. So there are 4 large data sets with thousands of patients that show that this approach, targeting toxic amyloid, can help patients, can slow the disease. And there's already kind of a lot of insight, on how what you need to do to make it more effective, to have it, safer and what have you. But again, the kind of the confirming, or the scientific approach, I think, has been the most dramatic. And again, for those who lived in drug development, there were decades of really no hope and no nothing. And suddenly, over the past year, it was not only the approval of Aduhelm for aducanumab, but also has been the emerging data from the other programs. So that's the first thing. The second, it really accelerated investments and support for the pipeline. And while there are no other disease-modifying treatments in phase 3 with positive data, and we have seen a lot of negative readouts from the anti-tau therapies and what have you, it really completely changed the perspective of the field, the perspective of the Wall Street, and so forth. And then finally, as we discussed a little bit earlier, there's a regulatory precedent for approval. There are endpoints, and there's kind of the data sets that actually will allow us, I think, the other products to move forward. Now, with that said, right, so one of the impacts specifically on the industry has been that following the Aduhelm accelerated approval, there are other programs that have filed for what's called the breakthrough designations. And recently, it's the Eisai's lecanemab that received the breakthrough designation, and the Lilly's donanemab as well. And the Lilly actually announced that they will also seek accelerated approval, with their filing actually starting this year. And just a few days ago, Eisai announced that they started their rolling submission for their approval. So what we're, again, looking at this, there are going to be, multiple options for patients, and I'll talk about the product profiles in just a second, but you can see that actually, following the aducanumab, there are at least 2 or 3 programs that could get on the market in the 23-24 timeframe. Just one comment on gantenerumab. So that's the only in this lineup that did not show positive clinical efficacy. They have shown effect on some of the biomarkers, but did not show clinical efficacy in their programs. So just one comment. So on the Wall Street, so the floodgates of funding had opened. We have seen it in the completely 180 degrees change of interest of investors on the Wall Street and the pharmas, really, because everybody who does not have it, the big pharmas who don't have Alzheimer's program, are turning to their BD guys and say, where's my Alzheimer's drug? And then also we have seen the one, which I consider very important biomarker, a company called Acumen was developing anti-oligomer antibodies, selective anti-oligomer antibody, has managed to go public and fairly successful IPO. So that is kind of the biomarker that even the hedge funds and these kind of risk advisors, the smart money, are following this. So if I go to the second slide. So the emerging pipeline, these are the drugs that have positive data, and it kind of summarizes, what we're going to be looking at actually the emerging pipeline and potential therapeutics. So, and I'll talk a little bit about the kind of the issues that have been dealt with and where we're trying to actually sort of improve. So you see the aducanumab and donanemab have very similar product profile. They have about 30% efficacy on cognition and function. As all the antibodies, they don't have to get to the brain very effectively. And as Duygu already mentioned, it appears that the selectivity for oligomers, the small soluble aggregates of amyloid, are the toxic species, not the plaque, as we discussed. And the selectivity of those 2 drugs is similar and not very good. That leads to both the limited efficacy, about 30%, but also the burden of the brain edema and hemorrhage of about 30%. Now, one of the critical pieces of insight into the safety and efficacy is the effect of the ApoE4 genotype. So patients with apolipoprotein E4 genotype have tremendously higher, actually much higher risk for Alzheimer's disease. They get the disease earlier with 2 copies of this gene about a decade earlier. And they also have shown in all of these programs stronger efficacy than the patients who do not have this gene, because they have much accelerated amyloid pathology, and also showed higher incidence of the side effects. So in the ApoE4 carrier population, it's about 40% in these 2 products, and a donanemab population. But if you look at the homozygous population with 2 copies, it's actually over 60% of the incidence of what we call ARIA, the brain edema, in these patients. So, and donanemab, again, is just completed the enrollment in their phase 3, and they're, again, planning to kick off the accelerated approval process very shortly. The best antibody is antibody called lecanemab, and the reason why it's the best is it's raised against the what's called Arctic mutation, which is the mutation that causes increased production of these large oligomers called protofibrils. That's the reason why it has better efficacy, and despite the fact it has poor brain penetration, the improved selectivity really translates to the better efficacy and about half of the side effects. Our approach is very different at Alzheon. We have an oral tablet that allows you to prevent the formation, stabilizes the monomeric form of amyloid, and prevents the formation of the oligomers. And because we are very selective for this, we don't interact with the plaque. We have none of the ARIA side effects. We have seen it in very large population of about 2,000 patients that we tested. So this is again, the emerging pipeline was the positive data, positive clinical data. And again, the kind of the pivots or the focus points are: number 1, on efficacy. Number 2, which really is driven primarily by the selectivity for these oligomers. There are a number of papers published on this now. The second is the ApoE4 genotype, both the efficacy and safety. It's actually segregated based on the genotype. And finally, there's been a lot of discussion about the cost. And we probably will talk about it later. the cost for aducanumab, why it's priced now at 56,000 per patient per year, would rise, to over 100,000 per year per patient with the cost of the infusion sets and what have you. So that has been one of the big points as well, and we would need to figure out, a cheaper way to actually treat millions of patients.
Yeah, no, I think that's right, and certainly I think the controversy around the price is well stated. It strikes me, Martin, that the FDA is going to be busy, right, with a lot of expedited approval requests over the course of the coming years.
They will be, and again, all these products have a chance to get on the kind of 23-24 timeline, and they will all are filing, so they are very busy. And again, I mean, the FDA has looked for approval for aducanumab and all of these programs, right? So they look at it as a class, and they will look at it actually through all in that consideration. So again, there will be treatments on the market again, and hopefully we'll be able to get some better. But again, they are very busy, and this is going to be a huge focus for the agency.
Yeah, absolutely.
Can I ask a follow-up question to Martin quickly? It's historically we also had some trials failed from clinical endpoint perspective, but they also showed biomarker changes in the past, but they never considered this accelerated approval path back then. Do you think there will be another chapter of going back and taking another look at some of those failed trials? And
that's an excellent question, right? Yeah, so the topic for this is not the biomarkers, but the other progress, what Duygu is saying, has been really understanding how the biomarkers correlate to efficacy. And there's been, again, tremendous progress, especially in understanding how the phospho-tau correlates with efficacy. The programs that have positive clinical data have effect on the phospho-tau. There are also very good data sets on clinical imaging, primarily to brain atrophy and what have you. So I would agree with you that they will likely actually go back and try to do that. But again, at the end of the day, the FDA would have to approve the drugs based on helping patients, right? So the biomarkers are tricky. Again, that was the only option for FDA to approve aducanumab. They could not approve it based on the data set because it was insufficient for approval, but on the clinical data set. But I think from now on, and also given all of this, the controversies and all of the discussions in every media you can imagine, there would be much higher, I think, bar for actually approving the drugs. And again, the helping patients is the only reason why we approve drugs, right? So it cannot be just based on the biomarkers or some kind of surrogate endpoints, especially as controversial as the plaque. The last point, the only thing that correlates with plaque removal is actually brain edema and hemorrhage, the side effects. And this is because you remove the plaque from the vessel wall and you make them leaky for plasma and blood. That's why you have the ARIA, brain edema, ARIA-E, and the microhemorrhage, the ARIA-H.
Yeah, but another thing I think is important in this regard is that the approval of FDA is conditional to the company running a post-marketing trial and showing actual efficacy on clinical endpoints. So
it's very true. Very controversial actually at this as well, because they have 9 years to complete it. There's no agreement on the trial, which would have to actually do that. And as you see, Biogen is already pivoting to Eisai's lecanemab, because they have 50% half of the economics. They co-own it actually with Eisai. So even in their presentations, you see they are highlighting in the fact that the Eisai data that showed lack of correlation with plaque removal and what have you. And again, the lecanemab appears to be the best antibody in the pipeline. It's the most selective, has the best efficacy and about half of the side effects. So again, there's a commitment to complete the study. I haven't heard the announcement that they are starting the program. And again, with the issues surrounding the launch, which has not been very as effective as they had hoped for, and the pickup, in the marketplace, I think there's a very clear, again, if you look at their analyst, presentations, they're already pivoting towards the lecanemab.
To add to the previous question, like we mentioned like a couple of times, so like with the whole assessment or approval of like this kind of treatment is all based on clinical benefits, and these are based, I think, right on ADAS-Cog and these kind of like assessments that are still on their not the most quantitative, right? They're still like subjectivity. They're like, can actually these kind of approvals be an opportunity to consider other markers or other imaging markers through the process from, is it the drug or the treatment actually modifying something on the biology? Can we see that on surrogate endpoints or in other markers through the process until it actually has impact on the cognitive functions? Kind of is an opinion / question to the group.
There's some precedence in multiple sclerosis, in our field of neurology and imaging. But again, I mean, you really need to there's a guidance from FDA and EMA, on how you develop the surrogate endpoints. The plaque removal does not fit that guidance, obviously. You have to have very compelling clinical data to support it in the large, completed studies before you actually make it a surrogate endpoint. And I think we're very far from that. And again, the risk, as we just discussed earlier, Duygu actually pointed out right at the beginning of this discussion, is that if you pick a wrong surrogate endpoint, you're shooting yourself in the foot, which really has been the case. And in fact, in scientific literature, the main point has been discussed has been, the lack of correlation of the plaque removal with the clinical efficacy, which shown in a number of, large studies with other programs.
One of the things as we looked at this emerging, one of the controversies was related, as we mentioned earlier, around the study data and whether the amyloid hypothesis endpoint was in fact borne out in that data. When you think about that and study setup in general, I mean, Paulo, maybe I'll address this one to you. Yeah, how and others to jump in. How should studies, trials be set up to avoid these issues, right? So that bearing in mind that we've got that ability to potentially be looking at more surrogate endpoints and potentially multifactorial things. How should we be setting these things up? How should people be thinking about this to avoid these issues?
I think like part of or related to like all the discussion about these trials is also like this ambiguity or the subjectivity or the lack of clarity of actually like what's the process, the design trial process, like and this lack of connection between the plaque reduction or not, and then the clinical benefits. So kind of back to my previous comment or my previous questions, like I think like it really stresses the need of introducing measures that are not subjective or less subjective, really to quantify that, like what we saw and what we're seeing on MS, that the clinical outcomes are more quantifiable, that there's like imaging or data-based markers that then can be correlated to clinical outcome. In the case of MS, like assessing the impairment on a motor scale, EDSS, is not the most objective for the range of different symptoms. So I think one point is really that, and what can help with that is like what we're seeing also a trend of data, real world data, wearables, some data that is collected like more real time and indirectly through the lifetime of the patient. It's like to really help in having a full picture of what's happening with the patient, with the person itself, and not based on like on basic or simple scales like this. So I think like imaging techniques like PET and others can really be stressed and to really automate. I think also that's the other point, to automate this, even the reading of images, to really be objective and reproducible and not like based on the manual assessments of that data and those images. So I think it's a mix of integrating data and analytics could help raise the confidence in the results.
So
couple things.
Yeah, please go.
Yeah. So as a researcher, like my point of view will be mostly from research perspective, but I do interact with clinical trials a lot too. So even within the research community, that there's this huge ongoing argument about the amyloid hypothesis. It's like there's of course no question that the buildup of amyloid plaques is hallmark of Alzheimer's disease. It definitely leads to this memory and everyday life disabilities. But to some, getting rid of amyloid plaque is critical to helping patients. But at the same time, that there's this other side of the coin that considering that downstream accumulation of tau and the neurodegeneration, those are the ones we know that leads to the actual symptoms. And those are the ones from like in vivo data, as well as autopsy studies, that we know those are the ones that truly correlate and predicts the clinical symptoms. The idea of getting rid of amyloid plaques probably should be done earlier, or it should be a stage or state dependent concept rather than imposing that amyloid hypothesis to everyone who might be on that Alzheimer's disease spectrum. So that should be something to be very mindful of. At what disease stage, which biological changes are actually driving and predicting each individual's progression at certain clinical and cognitive functions, and how can we leverage that information, or how can we bring the key biomarkers to truly understand subject-specific risks and subject-specific progression in the disease. It's very important to make these clinical trials successful. It's not just one fit all kind of approach for Alzheimer's disease. We already know that from research, and we already know that from our clinical trials. And when we look at the Aduhelm trial, there's not enough biomarker data actually to talk about its effect on downstream neurodegenerative changes. They did show some results from CSF biomarkers looking at p-tau levels, and but it was really a small sample size, not really giving us the enough confidence and enough power to really conclude if this is really a disease modifying approach. And so like definitely including biomarkers, but also being mindful of which biomarker is the key biomarker for that particular participant cohort, for that particular mechanism that the treatment's supposed to act on. Those are the things that should be included in my mind, that should be included in a study design for future trials.
Yeah, it's a great observation, and it's a real tension, isn't there, with personalized medicine, more focused and development in drug development, and the consequent high price associated with broader solutions, which clearly have not materialized for these diseases. It sounds like, Martin, you've got one thing to add.
Just following on your point, and what Juan think. Yeah, so what we have learned also is that Alzheimer's disease is a heterogeneous disease, that about a third of the patients that we thought have the disease, if you clinically diagnose somebody with Alzheimer's, you're about 30% wrong, right? So we have improved our ability to find patients who truly have Alzheimer's. We have defined what those patients are with what's called the ATN criteria. And then also what we have learned, and what really our pioneering research was really in the precision medicine, personalized medicine to Alzheimer's, because you really need to look at these different populations based on their risk, and in particular, actually the ApoE4 risk, because that's the after aging, the biggest driver of the disease. 2/3 of the patients have this risk, and the patients behave very differently, as we know. Now, coming back to the amyloid hypothesis controversies. Number one, there are hundreds of mutations and the hundreds of studies that actually kind of showing preclinical, clinical, and what have you, that this is really the only thing that have shown to date any effect on the disease course, right? And again, I mean, I wish we had something else, but it's not. The question is there still a cascade, or you need just impact on the amyloid to actually, slow or stop the disease progression? And this is open for discussion. But again, I mean, we have the big breakthrough is that we have large clinical data sets that support, all of the genetics and the other studies that show that if you interfere with the amyloid pathway, you can help patients and slow the progression. Now, I just want to say something, what Paulo was saying previously. Was one very interesting result of all these data from the late stage studies is that, despite of trying all these different composite scores and what have you, really the best performance was still on the gold standard clinical measures on ADAS-Cog, CDR sum of boxes, the ones that have been used for approval of all the symptomatic treatments for Alzheimer's, and they still appeared to be the most reliable. Now we need to, if we want to, have drugs, we need to have effects on the clinical measures. The biomarkers help find the right patients, to select them well and give us additional confidence, but we need that effect. And quite frankly, one of the biggest lessons from this as well, as has been, shown previously, we just need to complete the clinical trials, right? We can, the kind of the idea that you're going to save some money by cutting it short has just been the biggest problem, quite frankly, was the Aduhelm. If they completed studies, they probably would have gone through the regular approval, and we would have actually, much more compelling case and path forward.
Yeah, no, it's well said. It's well said. I want to bring Juan into the conversation. Juan, I'd love to hear your perspectives. This has obviously been a US and FDA conversation, but a broader implication, right? So, what's your lens? We'll make you the EU spokesman. What's the lens from the EU, and what's your thoughts on the discussion?
I'm speaking on my behalf only, but well, the EMA, the European Medicines Agency, shares the same goals and largely shares the same criteria than the FDA in general, generally speaking. So they harmonized a lot, and they tend to agree a lot. Of course, normally when a company files to these agencies, the decision is much clearer than not in this case of aducanumab, and we heard about the discrepancies and the controversies. So it's hard to predict what the EMA will end up deciding. It's about to decide by about the end of the year. One key concept and one key process that we were speaking about was the accelerated approval from the FDA, as opposed to the standard one. And in the European Medicines Agency, you also have this kind of mechanisms of expedited approval. And in Europe, we have the conditional approval, which also make in which drugs in which have shown not clinical efficacy in the standard process, and you might be relying on surrogate markers. So this is also a way that the EMA has in place, even though it's true that they typically use it way less than the FDA. So the FDA tends to use this kind of expedited mechanisms a lot more than the European agency. When there's discrepancy between the two agencies, normally it's because of, well, often it's because of that the clinical data submitted is not exactly the same, it's not regarding exactly the same way. So it doesn't necessarily mean that they have a discrepancy in the actual scientific discussion of the efficacy versus safety of each drug, but that may also happen. So we know about in difficult cases, for example, in Duchenne muscular dystrophy, where trials are difficult because a few patients are difficult to control for placebo. Often the FDA and the EMA have had different opinions with the same data, with the same trial data. Then what is really different, the other thing is that what they might be differing more likely is in the indication. So even the drug is approved, what is the exact indication, who might be eligible for that drug, and that also is a surprise for me in my case, that all these trials have been performed in amyloid positive individuals. As Martin was saying earlier, about 1/3 of the patients with the clinical diagnosis of Alzheimer's disease do not harbor amyloid in their brains, and you can call it the way that you want. I mean, this is our amyloid removal drugs. So if you don't have amyloid in the brain, there's no chance it having a clinical benefit. So it might well be that the European agency might approve the drug in the same way as the FDA, but with a different indication. And finally, what is really different in Europe from the USA is the reimbursement. Because, well, in the US you have Medicare, Medicaid, or in the end, insurance companies will pay for the drug. In Europe is the national healthcare systems, and then really goes to the national level. So it's not a European approval. So then the health and technology agencies that will evaluate the cost efficacy of the drug are at national level, and even the payers might even be at the regional level. So even if the EMA approves the drug in all Europe, that includes the UK still, then reimbursement is really fragmented and might be really difficult to predict what the different countries or even regions might end up doing.
Yeah, I think that's well said, and also comes back to the point you were making earlier, Juan, wasn't it, about patient and advocacy in this area, and just being really clear and crisp around what these things do and who will be eligible, because let's face it, if you're someone who has this diagnosis or a family member with this diagnosis, you are desperate for something, and we need to make sure that the right patients are getting the right treatments and that it's accessible for all, right? I think that's the key point. Duygu, I'll shift to you in a moment, and a related question. I want to just in the last 5-10 minutes or so look forward a bit more. So with all these developments, right, and we've had a lot of discussion around the nature of this approval and the use of surrogate endpoints versus clinical endpoints, and particularly with these diseases. How does this make you or your colleagues think differently when you consider research? When you think about the way you shape that agenda and execute, does it change the way you do things?
That's a great question. So first of all, it's like, even though we are like tearing it apart, there's definitely huge excitement, right? It's the first drug that is approved in a new category, and we know it's going to open many new doors, bring many new opportunities. So we definitely see that as a step forward in progress in dementia research as well. And also the nature of the approval, I think it's this recognition of how biomarkers are so critical in Alzheimer research and how critical they are for us to understand the disease progression and then the response to treatment. So for one thing, we know neuroimaging as well as fluid biomarkers are going to be a key part in these trials, because as many of us have been mentioning, it's an amyloid clearance mechanism. So we really need to identify the right individuals for this kind of treatments. It's not going to work for everyone. So it's that patient stratification and matching the right patient with the right treatment. That's where we will put, we're going to keep putting lots of effort on biomarker discovery, detecting the underlying pathology, as well as predicting and monitoring how each individual is progressing based on their biology to properly match them to different treatments. So I think and we're already seeing that traditionally we've been using amyloid PET imaging, CSF, like lumbar punctures to detect Alzheimer biomarkers in living individuals. And now we're in this area of huge excitement around plasma biomarkers. These are like blood biomarkers. They're scalable. They're cost effective, and with those biomarkers we can actually reach to many different populations. It's just because they're scalable. So we are always going to be in search for those scalable, cost-effective biomarker approaches to detect and monitor AD pathophysiology and the progression of the AD, and bring those to the clinical trials to facilitate not just only selecting the participants who's going to benefit from the treatment, but there is definitely more going to be more emphasis on how to discover the true biomarkers, surrogate measures that are truly monitoring the disease progression together with the clinical outcomes. Because as Martin said, it's at the end of the day, all we care is how the patients are improving. So our surrogate measures, our biomarkers should reflect that improvements in an objective way. We need to put more effort on developing those biomarkers and truly validating those biomarkers to avoid such discussions in the futures.
Yeah, no, I think that's well said. That's well said. Let me shift a little bit to, if you like, the pharmaceuticals, the biopharma industry itself a little bit. And let me start with you, Paulo, and we'll expand it to Martin and Juan. You know, with all of this going on, and this is still very fresh, and we're still processing this shift that we're seeing here. But what should biopharma companies be doing now, right? To, if you like, to lean into this change of direction and potentially get things moving more effectively to get more therapies into the marketplace.
I think like overall, like we all will cover like some of the points, right? Like this you know, the FDA this approval like just brought flexibility and excitement back into the field. Investments, you know, the pipelines that were shown. So like there's more and more, you know, reigniting that whole field. And I think, and that, like Duygu was mentioned, that also, you know, opens, you know, underlying the need of objective biomarkers, objective data that, you know, helps to connect the dots, like from, you know, what are the mechanisms, what is the treatment doing, and then what's the benefits for the patient. So I think that also, like was mentioned, like it opens the door, and I think like the biopharma should be doing that and are doing that. That is to look at all this, you know, huge amount of data that have been collected in stale trials, like things that didn't seem that there were any benefits or you know, very small benefits, like revisit that, because now we have, you know, great new technology, new analysis, AI, etc., that can really sieve or analyze back that previous data, and then, you know, helping either in stratifying patients, figure out like actually from this trial that failed, but did it fail really like the same way for everybody, or it's like a specific subgroup? What's different in that subgroup? Could then we target like the new treatments for that subgroup? And I think like it's really like totally underlying the comment that it is an exciting time, and I think you know, it's for us, for everybody to really help in taking that to the next level. And in the end, again, not forget that is all to improve, you know, patients and, well, their families as well, lives. So I think it should be doing that.
Yeah, 100%. It's what it's all about. Yeah, it's interesting. You and I know that a number of our customers, QMENTA's customers, are actually coming to us saying, "Hey, we've got these data sets, and we just don't think we've extracted the value from those yet." And so being able to kind of dust off some of these things and take a fresh look in light of this shift of approach, I think has a lot of value, exactly as Duygu was saying earlier as well. Martin, any views for you? I mean, you already spoke a little to, you know, the pipeline and the investor perspective. Super exciting to see money coming back into neurodegenerative diseases. Any things that you would, any advice or direction you would give to the industry and what they should be focusing on now?
Maybe 3 points, and if I can bring back that pipeline slide, that slide number 1, Chris. So the first thing is, you know, and this is again tectonic change for everybody in the Alzheimer's space, with respect to there is a hope, there's a treatment, and there's a pipeline. I think we have to really appreciate the fact how tremendous impact this is on the patients, on the physician community, you know, on everybody in the industry, because there's a clear path forward. And again, there will be a pipeline. The specific, you know, implications long term, you know, the aducanumab or the Aduhelm launch has not been as successful as Biogen has hoped for, and I honestly, I doubt that this is going to be the standard of care as we would see it that way. There are other and better drugs coming down the pike. and I think they will have a chance to actually become the standard of care. And again, in the 23-24 timeframe, we will have an options, and really the focus needs to be here on the product profile. We can improve we need to improve the efficacy, the selectivity of these agents, and the safety. Again, the safety is paramount. There have been already reports, from even from the launch Aduhelm, some of the side effects in these. So that has to improve. The second is the pricing. There's just no way, we can have 100,000 per year per patient, for millions of patients that need this kind of treatment. It's just not going to work, and there's going to be huge pressure everywhere. And in Europe, it's just unbelievable. It's never going to happen. I mean, as you may know, Europe, the NICE Institute actually never allowed Aricept to be actually reimbursed, which is the cheap symptomatic. Now, the last thing is, which really not to forget is the really where do you need to intervene in this disease process? But people forget that what we see as a clinical stage is the terminal illness. You have 20-25 years of brain damage that's evolving, and the brain is very adaptive, actually, and able to compensate for that. But really, the last few years, when you have the clinical symptoms, is where we currently use these treatments. We need to move earlier. We need to move into preventive paradigm. We need to have, safe, and we need to have oral drugs, that actually would be able to intervene early. Because again, the main risk for Alzheimer's is age. Every 40-50 years, all of us start having these changes. And if we talk about 335 million people with the clinical disease, there are at least 10 times as many that have the changes in the brain, which now we're talking about hundreds of millions of people who need this kind of treatment way before, they have the clinical symptoms. And there's an opportunity now to intervene early and actually make that happen. That will change the disease.
Yeah, no, well said. I think all of us can align on some of these points, particularly around the accessibility of this treatment, and exactly as you say, getting towards prevention rather than care. And as someone who's has a loved one who's suffered from this disease, as many of us has, and it touches so many people, all of us can just hope and pray for that. We are out of time, and with apologies to a few of the questions that came in there, I will try and follow up with those separately. But I do want to extend a massive thank you to all of the speakers, to Martin, to Juan, to Duygu, and to Paulo. Did an outstanding job in packing this incredible topic. If this one's going to run, I'm going to run. Just amazing stuff. Thank you for joining us. There'll be a post-webinar survey that will go out. Please give us your feedback, give us thoughts on things that you'd like us to cover in the future. There will be a recording of this that we'll share it with all your friends and family, and enjoy. With that, we'll wrap that up. And again, with thanks to all of you, great job, and we'll see you all again soon. Take care all.
Thank you.
Thank you.
Thank
Bye.
you. Bye-bye.